Hello, the session is about the applied or the applications of microbiology in various fields. In the previous session, we noted about the different levels, why this microbiology is important. So let's see one by one. Microbiology has got its applications in various fields like environment, it has got its application in medical field, agriculture field, in warfare, so many things. So first about the applied environmental microbiology. That is the application of microbiology in the environmental applications. And the first one is bioremediation. Bioremediation means the application of microbial organisms to control pollution. So the microbial organisms or microbes will be applied to the polluted or contaminated and thus the waste materials are degraded with the help of the organism. So in large amounts the microbes will be introduced into a particular area where the pollution is very high. That is known as bioremediation. And bioremediation is of two types, in situ bioremediation and ex situ bioremediation. In situ bioremediation means applying the microbes in the particular area where the pollutants are present. That is, the microbes are applied in the natural habitat itself where the pollution is very high. And a common example for that is Pseudomonas putida, that is the superbug. Pseudomonas putida or the superbug is used in oil spills. So they are oil eating bacteria and they will degrade the oil very fast, which is spread over the oceans. And ex situ bioremediation means if the pollutant is or the degrading material is taken from its site to a controlled conditions and the microbes are applied on it. That is known as ex situ bioremediation. So that is in situ and ex situ bioremediation. And of course, we are very much aware of the fact that it has the application of microbes in composting. That is actually composting is a method of bioremediation itself, that is treating the biodegradable waste. Composting, the important thing is you can only degrade those materials which are biodegradable. The natural microbes can degrade only the naturally occurring or biodegradable materials such as plants or animals. That is what biodegradable materials. And of course, there are few microbes which can degrade the materials which are not biodegradable. There are few bacteria research are going in that or going on in that field where the microbes can degrade the plastics too. So usually or common practice which we observe in and around our household is that the microbes can degrade the biodegradable waste, that is known as composting, and this is and large quantities of composting will be done in controlled conditions. We have biodegradable this waste processing plants and all uses the microbes for degrading the waste materials. Then next is biostimulation. Biostimulation means the the beneficial microorganisms are grown and that is used for controlling or degrading the contaminant. So biostimulation is actually defined as the release of nutrients, oxidants or electron donors into the environment to stimulate the naturally occurring microorganisms to clean up the environment. So this stimulation means uh, stimulation, biostimulation means certain stimulants. Biostimulation means certain chemicals are applied onto these fields which can enhance the growth of the microbes. That is known as biostimulation. Biostimulation means certain chemicals will be applied to the field or certain nutrients. The biostimulants will be nutrients which are given to this microbe so that it can efficiently degrade this waste, and that is what is known as biostimulation. Then next is bioaugmentation. Bioremediation is over, composting, and next is biostimulation. Biostimulation, this is L, sorry. Biostimulation. Biostimulation means the nutrients are supplied to those microbes which are involved in cleaning up activities. That is known as biostimulation. So, next is bio-augmentation. Bio-augmentation is actually the addition of special microbes which may be or may not be a natural inhabitant and that satisfy the requires of the cleaning. Bio-augmentation means addition of more microbes, addition of more microbes into a specific area and that is known as bio-augmentation. And for bio-augmentation, for example, you have a particular area or a particular tank in which a pollutant is present. You need to degrade it. So at that place, there may be 10 microbes. This is actually hypothetical data. 10 microbs. We will add 20 more microbes to that area from we will collect the extra microbes from another polluted area. That is what is known as bio-augmentation. Bio-augmentation means more microbes will be collected from another site and will be introduced into the specific site. That is known as bio-augmentation. So for this, the commercial culture tanks will be there for microbes. You can collect that microbes. That is why we are given some, for example, for composting and all, some we get this things in bottles. In that bottles, they will say you need to spray only this material on the biocompost. And what happens is that in that bottle there will be microbes and they will degrade, they will multiply and they will degrade the waste. You have a tank of waste and for degrading that waste you will be given a vial or a bottle of liquid like substance. You will pour the liquid on this waste and after some times this waste will decompose. Actually what has happened in that small bottle which you have provided there will be microbe and that microbe will multiply on this biocompost at this compost and it will degrade it. That is what is known as bio-augmentation. So bio-augmentation means addition of more cultured microbes onto a particular area to enhance the degradation. Next is biofertilization. Biofertilization means actually we have not many nitrates or the fertilizers present in the soil in its natural condition itself. So continuous agriculture practices, we are very familiar with the term biofertilizer now. For the continuous or sustainable agriculture practices, the usage of biofertilizer is very important because that will improve the health condition of the soil itself and also the plants, the plants and the food products which we get from the plants. So that is what is known as biofertilizer. And this biofertilizer, this can be compensated by culturing the free nitrogen fixing microbes. And the common nitrogen fixing bacteria are present in the soil itself. That is, acetobacter is present. But what happens is that in the field, to enhance the fixation of nitrogen. You have learned the nitrogen fixing bacteria and all. Natural condition itself, there are some microbes present in the soil. But to increase the content of nitrates in the soil, more nitrogen fixing bacteria will be introduced into the field. And that is a biofertilizer. For example, one example is Anabina azola and Anabina azola, and that lives in symbiotic association with a free living water fern that is azola, and that is used as a nitrogen fixing cyanobacterium. So biofertilizer means biofertilizer, I'm talking about the microbes. Biofertilizer means naturally nitrogen fixing bacteria are there, but we add more nitrogen fixing bacteria to the soil to increase its fertility. That is known as biofertilization. Now, next is so these are the different things which we learn. But then next is the sewage treatment. Sewage treatment is the sewage is the waste. That is the treatment of sewage is a very important or a major concern in the urban situations. We are all aware that the sewage treatment is very important or a matter of discussion every day, every hour, because nobody can degrade the material or the waste products which are coming from their house itself. So sewage is a major concern mainly in the urban condition rather than the rural condition where people live in multi-story flats or where people live in small houses within small area and all. And so though that is of a great concern, and that is an important field of for sewage treatment, the microbes are used extensively. And the sewage treatment involve different steps, that is pre-treatment, primary treatment, secondary treatment, and tertiary treatment. Pre-treatment means first when a sewage is collected and piped into the treatment plant. For sewage treatment, we have some treatment plants. One of the common treatment plants which we had in Trivandrum city, we had a sewage treatment plant, but now it is shut down due to certain reasons. And the sewage will be collected from different places or household conditions, or will be collected and will be brought together in a plant. That is, then the sewage is before treatment, it is subjected to preliminary filtration, that is known as pre-treatment. So in pre-treatment means the sewage is that pre-treatment means it is subjected to sedimentation process. It is subjected to sedimentation process before the treatment begins. So first sewage will be taken to the plant. It will be put in water and it will be allowed for sedimentation. And after sedimentation, the floating material will be removed and the solid material will settle down. So in pre-treatment, the first thing what we do is it will be put in water, and that is the preliminary filtration. So the heavy materials will sediment and the light materials will float. And so the floating materials will be removed fast and the solid material is then further processed. So in the next step, the sewage is pumped into. Next is the primary treatment. So pre-treatment is just isolating the floating and the solid component. And here in this chapter, you need not learn about the detailed process. You just learn the different steps of this. So primary treatment is the sewage is piped into large open tanks and again it is set for sedimentation and the organic suspended materials will be separated. In the primary treatment, the organic material will be separated and this organic material will be subjected to further treatment. And in order to increase the efficiency, the substance flocculating substances such as aluminium and iron sulfates are added, and the solid material that settles down is called the sludge. Hmm? Sludge. So primary pre-treatment, floating, sedimentation separated. Primary treatment, again the sedimentation itself, but by adding certain chemicals to improve the isolation of the degradable material, and that part is known as sludge, and the one which we get is by the treatment of this flocculating substances is called as sludge. Then next is secondary treatment. So the sludge and the liquid is separated into different tanks. After primary treatment, the sludge or the one which has sedimented and the watery component will be separated into two different tanks. Then the secondary treatment of the liquid and the solid phase, and actually from this phase, actually the secondary treatment involved the role of microorganisms. The microbes are used in the secondary treatment. See, from this step only, the microbes are used. Microbes are used in the secondary treatment. So, the microbes which are used are Bacillus subtilis, Pseudomonas species, cyanobacteria, and some other organisms too. And those bacteria which can live in aerobic and anaerobic conditions are used in secondary treatment. And while using this microbes in the secondary treatment, additional chemicals are also added to it, and these additional chemicals are actually meant to maintain a constant pH because microbes need a specific environment to live. otherwise it will die. So that that materials are called the probiotic chemicals. So in secondary treatment, microbes are used for the efficient sustenance of microbes. Probiotic chemicals are also used along with it. And the then it will be undergo. That is the proper by providing this proper aerobic and anaerobic condition. And usually the anaerobic conditions are prepared by these microbes, and the process of microbial action on the sludge will take place. That is, the sludge will be digested. Sludge will be digested by these microbes, and the sludge will be converted into a agricultural fertilizer. So sludge will be converted to a fertilizer. So you can use that as a fertilizer. Sludge will be converted into fertilizer, and that is happening in the sewage treatment with the help of this microbes. Now, next is, so in secondary treatment, what we did, we got a solid or a semi-solid substance, that is sludge, after primary treatment. In secondary treatment, microbes are used. Microbes which are used are Bacillus, Subtilis, Pseudomonas, Cyanobacteria, etc. And usually the anaerobic conditions are prepared by these microbes. And for probiotic chemicals are added for maintaining a good culture of microbes. And the microbes will eat, or they start feeding on the sludge, and the sludge will be converted into a fertilizer. Now, the last step is the tertiary treatment. Tertiary treatment means the liquid phase. We have a liquid phase. The solid phase was the sludge, and we have a liquid phase too. And the liquid phase is subjected to further treatment in the tertiary treatment. And the liquid is subjected to the anaerobic treatment in the secondary stage. Then, as a result of that, it is treated in oxidation ponds and encouraging the growth of aerobic microorganisms. The liquid treatment or the liquid part is subjected to treatment with the aerobic microorganisms too. And as a result, these microbes will eat up those biodegradable things in the water, and then this water can be reused again either for watering the plants or in such things. And there are certain conditions where it is purified and again used back also. So these three are the different treatments. Sewage treatment. Sewage is the waste which is collected. It is subjected to pre-treatment, that is purely isolation of the floating and the sedimenting products. It is subjected to primary treatment. Then secondary, after primary treatment, you get a sludge, and in the secondary treatment, microbes are used. Tertiary treatment, the water component is further purified with the help of this small microbes, and the purified water will be sent back to the into the natural water bodies, usually away from the human habitation, and there are places where this is used for agricultural purposes too. Then next is applied agricultural field. Microbiology is used in agricultural field, and let's see which are the which all sectors it is used. And the main place where it is used is, of course, the bio fertilizer itself. We have learned about, just mentioned about it, that is the nitrogen fixing bacteria are used to enhance the fertility of soil. So biofertilizers are of different types: nitrogenous biofertilizers, phosphate biofertilizers. So the nitrogenous biofertilizers are the free living nitrogen fixing bacteria will be introduced more into those fields, and the bacterial inoculants, and that is what I said. Inoculants means a culture will be given. Inoculants will contain rhizobium, acetobacter. So biofertilizer means in small bottle you will be given a liquid like substance, and you will be asked to spray it on the soil. And in that small bottle you will be having rhizobium, acetobacter, azospirillum, cyanobacterium, etc. And that is usually used in between the crops. Crop rotation after every harvest or after two harvests, preferably this can be used. Then phosphate biofertilizers, and the phosphate biofertilizer improves the phosphate content in the soil, and phosphate biofertilizers used are Bacillus megatherium, Pseudomonas, striata, etc. are the common phosphate fixing microbes that are used. Then biopesticides. Biopesticides means that it is very important. In biopesticides, BT, that is, we have biopesticide is BT toxin. Actually, BT toxin means BT. It is a short form of Bacillus thuringiensis, which is a bacteria. BT stands for Bacillus thuringiensis, and Bacillus thuringiensis produces a toxic substance, and this toxic substance will kill the lepidopteran insects. Mhm. So this Bacillus thuringiensis, if it is sprayed on the plants, it will attack or it will control the lepidopteran insects such as moths and butterfly. For example, you have cabbage fly. Cabbage is usually eaten by a butterfly, that is the cabbage fly, and this cabbage fly can be controlled by spraying this Bacillus thuringiensis on this specific plants. And when the larvae, for example, you are spraying Bacillus thuringiensis on the cabbage plant, the butterfly larva will feed on the cabbage. When the butterfly feed or start feeding on the cabbage leaf, what happens is the larva will die as soon as it reaches its body. So that is what is known as biopesticide in the aspect of microbiology. There are other biopesticides too. I'm talking here about the microbiology. So microbes which are used as biopesticides are Bacillus thuringiensis. It produces a toxin that is called Bt toxin, and usually it is efficient in controlling the lepidopteran pests such as the butterfly or the moths. Then next is bioinoculation. Bioinoculation, this is what it is very important. Then bioinoculation means the culturing of microbes. Bioinoculation means that is known as bioinoculum. 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 Culturing of microbes. Bioinoculation means suitable microbes are cultured and their spores are collected and stored in a inoculum. It can be a liquid. That is what I said. Usually in a small vial or something, you will be asked to spray on the field or on the compost. So a bioinoculum contains cultured microbes, spores, and they will initiate the population growth when it is applied into a particular area. So, bioinoculum can be defined as a culture containing viable cells which functions as the initiating population of parental cells capable of multiplying in a system to which they are applied. Example, that is why it can be applied. Inoculums will be provided in small bottles in liquid condition itself. Either they are for the nitrogen fixing bacteria can be given, or the biodegradable the capacity, those microbes which are having the biodegradable capacity are also given, and that is known as bioinoculum. It is a small culture containing the microbes that which are capable of initiating or propagating the multiplication of that group of microbes. That is known as bioinoculum. In a small bottle, microbes will be given and it will multiply and increase in number and efficiently will work on the field, either on the agriculture field or on the this biocompost or anything. Depends on its application. So the advantage of the farmer by using this bioinoculum is that it reduces his dependency on the chemical fertilizers. He need not depend on the chemical fertilizer. A small amount of inoculum is enough. But using of inoculum helps reducing the usage of chemical fertilizer as well as help in reducing or mitigating the cost of production. Mitigating means to increase the cost of production of the plants will be in a controlled manner with the use of microbes. So that is advantages for the farmer. The next is that's all about the application in agriculture field. Next is application in industrial microbiology or in the industrial sector. And this microbes has got wide application in the industrial field. That is, it is applied in the metabolic processes which is happening in the body of the microbes are used in the industry. What actually makes the microbes very what friendly for the industrial units are, they are called the mini chemical factories. See, it's actually a small microbe itself. And now what's happening also is like that. This COVID-19 virus or something, it is very small, but the work which is done by it is very high. So microbes are usually mini chemical factories. And they have the capability to grow very fast, and they can be, these are the characters why microbes are used extensively in the microbiology or in the agriculture or in the medical field. In so many fields it's used. It is a very tiny organism. See, it is invisible to your eyes, but it is working as a mini chemical factory, and it can grow rapidly. Then it can be cultured in large quantities. See, if you need to take a big organism from one place to another, you need to take a big a cage or something like that. But carrying this microbe is very easy. You can need to put in a small vial, you can put in a small tablet form or even smaller size than the tablet too. So the culturing in, you can culture the microbes in large quantities, it grows rapidly, then it maintains the physiological consistency after each culture, and it has the ability to carry out the transformation from the culture to the environmental conditions. These are the reasons why microbes are used. And next is the products and microbes which is involved in the industrial microbiology are, of course, first one is the organic chemicals. Organic chemicals, that is, the microbes are employed not only yeast but bacteria such as, and the microbes which we use in our daily at home itself, we are aware about the lactobacillus, then about this what yeast, etc. We are very I mean, at least in 2 days or 3 days we are depending on these microbes itself with our knowledge. That is important. So the beginning of the industrial microbiology started with the production of ethanol. Ethanol is used in the brewery industry. Ethanol was produced with the help of this yeast. So yeast has got a very important application in ethanol production. Then the microbes which can efficiently ferment are the Zymomonas mobilis is another microbe which is another microbe which can efficiently use it. Then other microbes used are the lactobacillus, then citric acid production, the bacteria which is used is acetobacter. So many microbes are there. For the lactobacillus is produced, lactobacillus help in the production of lactic acid. Citric acid is produced with the help of acetobacter. Then acetone, ethanol and butanol are produced with the help of this Clostridium, acetobutylium, yeast, etc. itself. There are so many microbes which are used. Then next is single cell proteins. That is, the microbes which are rich in proteins are called single cell proteins. For example, for means that single cell will contain a large amount or a lump sum amount of proteins. That is called single cell proteins. Examples are spirulina maxima is a common example which is having this, which is used as a single cell proteins, and they are used as nutritious food instead of food supplements. So because some microbes have large amount of proteins in its body, so they are used as supplements of food. That is what is known as a single cell proteins, and so they are cultured in large amount. They are dried and packed for using as the health foods or additives. And the common examples are the algal species, that is spirulina. We are aware of the spirulina is used by the space astronauts, or the astronauts use this spirulina maxima as a food because it can be dried, it can be stored for long period, and they have a rich amount of protein also. Say they are used as food supplements by the astronauts. They are called single cell proteins. Example spirulina, then acromobacter, then cellumonas. Then next is biodegradable plastics. And in today's for people are after for long periods people are after this plastic how to degrade it and all, and some solutions has come from the part of this microbes too. I said earlier too, there are some plastics which are biodegradable, and the common example which we know are the plastics which are produced from corn. Plastics that is biodegradable plastics are produced from corn. Biodegradable plastics are produced from tapioca. Biodegradable plastics are produced from by the mixture are made with the by the mixture of corn as well as the rice starch too. That they are the common biodegradable plastics. But some plastics are manufactured with the help of the metabolites that are produced from the microbes. Certain bacteria will produce some metabolites, and these metabolites are used in producing plastics. And the bacteria which are involved in this are lactobacillus, then alkaligens and pseudomonas. A species of lactobacillus, alkaligens and pseudomonas produce some metabolic substance from its body, and these metabolic substances are used in the synthesis or production of biodegradable plastics. But the limitation of this biodegradable plastic is with the enormous number of population, it is very difficult to meet the need of the large population with this biodegradable plastics produced by this microbes and also the natural products too. That is a limitation. And the cost of production is also high for this biodegradable plastics which are produced from the microbes as well as from the natural products such as corn starch and all. Then next is the bioreactors. Bioreactors are mainly, here you can see, that is in a controlled area or in a controlled vessel, the microbes are cultured. Simply bioreactor means a culture, a controlled vessel in which the culturing of microbes are done. That is called bioreactor, and it is used in industrial microbiology. That is, I said, industrial micro bioreactor is a system in which large number of organisms or microbes are cultured in controlled condition. For example, you need this nitrogen fixing bacteria. Nitrogen fixing bacteria is used by the farmers. So in this bioreactor, this is a typical model of a bioreactor. In a vessel, the nitrogen fixing bacteria will be allowed to grow or culture in this vessel, and based on the need of people it will be transported to its site by the it will be transported to its site by the in small vials, that is known as bioinocular. In this vessel the microbes will be cultured, it will be transferred to small bottles and will be sent to regions where they are used. Either it can be in a composting plant or it can be in a agriculture field. That is what is known as a bioreactor. Bioreactor means a control condition in which microbes are cultured. Simply, that is bioreactor. And these microbes are extensively used in the mining field also, and that we have learned in the previous sessions and all. That is, it is microbes are extensively used in mining, that is bioleaching or microbial leaching, is the process by which the microorganisms are used to dissolve certain elements and to isolate certain elements. For example, Thiobacillus peroxidans is a bacteria which is used in the iron mining, and in that field from the oxidation, they help in the oxidation of iron sulfide. Thiobacillus peroxidans, then Bacillus megatherium, then Thiobacillus thiooxidans. There are a lot many microbes which are used in the mining field to separate the metal from its natural ores. Natural ore of copper, iron, manganese, gold, etc. is present. In the natural condition, the microbes are used to separate the metals such as gold or this copper or iron or manganese from the natural ore. In that field, the microbes are used. Example is Thiobacillus peroxidans used to separate iron from its ore. So it is used in mining also. Next is the application of the microbes in medical field. So the advantages of using microbes in industrial applications are, I said before, they are very small, it can be cultured easily, and the metabolic products can be used. Then pollution is very less, comparatively the pollution is less, and the products which are produced by the microbes can be recycled. That is the importance, or that makes the usage of this microbes in various fields. The byproduct which is produced by the microbes can be recycled. And the cost of production is also very less. Let's see in which all field the how it is used in medical field. The In medical field, of course, microbes. They and many microbes cause diseases, and many microbes help in controlling these diseases too. So, applied medical microbiology is it is used in diagnostics, which helps to find the bacteria or the microbe which is responsible for a disease or certain condition. That is many microbes, and you can see that is this coronavirus causes COVID-19, and not many. This what you have what this what chicken pox, viruses and bacteria cause many diseases among humans as well as animals. That is HIV virus is there, then staphylococcus, then this bacilli. There are many microbes which cause so many diseases in the field. And even though these microbes cause diseases for humans, the products which are isolated from the body of microbes itself is used to control this microbe. What happens is, in medical field, what is done is usually antibiotics are given to control the bacteria. The diseases which are caused by bacteria is controlled by giving antibiotics, because antibiotics act against bacteria. Then most of the present day antibiotics are actually the metabolic products of microbes itself. Antibiotics are the metabolic products which are produced or in the body of these microbes itself. And once this antibiotic is isolated from a microbe, it is used for controlling the disease. And the more than so many antibiotics are used. That is, the common antibiotics which are used are the streptomycin, then penicillin, some of the common antibiotics which are used, and the different strains of streptomyces bacteria produce different antibiotics such as amphotericin, chloramphenicol. These are the common antibiotics which you might have heard about the terms itself. That is chloramphenicol, neomycin, etc. are the antibiotics. Neomycin, maybe you might have, that is ointments and all. If you read on the antibiotic ointments, you can see this neomycin, which is a very mild antibiotic used in the eye infections and all. And this neomycin, amphotericin, and chloramphenicol are the antibiotics which are produced by the microbe Streptomyces. And Streptomyces is a bacteria which is very common in the soil. It is a soil inhabiting bacteria. So what I said is, even though microbes cause diseases, that is of great concern in medical field. To control that microbes, antibiotics are used. From where are we getting these antibiotics? We isolate the antibiotics from the body of the naturally occurring bacteria itself. And the bacteria which is extensively used in the production of antibiotics are the Streptomyces, which is a very common microbe in soil. It produces amphotericin, chloramphenicol, neomycin, etc. Many other examples are also, that is the fungi, that is Penicillium notatum is a source for penicillin, and penicillin is actually the first antibiotic that is used in medicine. So it is produced from the fungi, that is Penicillium notatum, that is microbes and antibiotics. Then epidemiological diseases, sorry, only one A. Epidemiological diseases. Epidemiological disease means a disease which is spread in a area. That is epidemic, means it is transmitted, it is in an uncontrolled way. And one of it is, it will become a pandemic too. And that is what is happening now, that is this COVID-19 too. Then this what microbes are actually used to prevent the epidemiological disease itself. So one thing common is you produce vaccines. Vaccines are vaccination. What is used is the non-virulent strain of that bacteria. For in here in COVID-19 case, what we are using is there are different vaccines available. Some vaccine will contain the less virulent strain of. It's not a virulent strain, means disease causing. Less virulent strain of this COVID-19 virus itself will be injected into the body and antibodies will be produced against this virus. That is vaccination. In the field of vaccination, the microbes are used in case where the epidemiological diseases happen or a pandemic situation arises. Another field in case of this medical microbiology is the social and preventive microbiology. That is, the applied microbiology involves the field of social and preventive medicine. It involves the monitoring of environment. That is very important. A hygienic condition has to be maintained. Monitoring of the environment in which people live to ensure that they are free from the pathogens. That is, in a place there should be proper sanitation, proper usage of toilets, then proper treatment of waste materials, sewage treatments should be good, etc. And that should prevent maintaining a hygienic condition in a place where you live will prevent the sprouting of this uh, bacteria which are harmful to humans. And the transmissible diseases which come in those areas are cholera, typhoid, malaria, chikungunya, then diseases like plague, polio are some of the diseases which you can keep away if very good hygienic practices are adopted. Then vaccination, we have mentioned that earlier itself. There are different vaccines which are produced, that is common, which are given to the small babies, that is BCG, MMR vaccine, and now we have different vaccines like Pfizer, Covaxin. Then we have the Covishield, we have Sputnik. Not many vaccines are there. Then the application of the microbes or microbe in biotechnology. In biotechnology involves the application of scientific and engineered principles for the processing of materials. That is how biotechnology, that is techniques are given more importance. And in biotechnology or in a biotechnology lab, the scientists or the people or the researchers who work on this, they will be handling lot many microbes for the, because in the laboratory condition itself the production of many proteins can be done, and that is done with the help of these microbes. And the common example is one plasmid, that is a extra chromosomal material. Plasmid is a circular genetic material. It is extra chromosomal. And with the help of this extra chromosomal plasmid, gene transfer is made efficient. Plasmid, you can see if this is a bacterial cell, there will be a genetic material and you have another circular material, and that is known, that is called a plasmid. And this plasmid is used in biotechnology for transferring the genes from one organism to another. Then, when this microbe is genetically engineered and cultured to produce pharmaceuticals, and there are microbes which can produce insulin, there are microbes which can produce some other products, and that is how it is used. So, these plasmids are extra chromosomal genetic material which is present in the bacteria, and they are used in the transferring of genes from one organism or one microbe to another. Then this Taq polymerase. Taq polymerase is actually the polymer. This Taq polymerase is an enzyme which is used in PCR or polymerase chain reaction. And the importance of this Taq polymerase, the importance of this Taq polymerase is that it can withstand very high temperature. Taq polymerase is isolated. Taq polymerase is the enzyme which is isolated from the bacteria Thermus aquaticus. So this Thermus aquaticus is a bacteria which can withstand very high temperature, and Taq polymerase is an enzyme which is isolated from this Thermus aquaticus. And this enzyme is used in PCR or polymerase chain reaction to make millions of copies of a single DNA sequence. So, in a controlled laboratory condition, you can produce more amount of DNA with the help of this Taq polymerase. Another thing which is used in biotechnology is the restriction endonuclease. And restriction endonuclease, as you are aware, maybe aware, that is restriction endonuclease are those enzymes which are used to cleave the DNA strands at a restricted site. Endonuclease means inside the nucleic acid sequence, certain cut is made. That is known as restriction endonuclease, and this restriction endonuclease are commonly called molecular scissors because they cut the specific DNA sequences. So in biotechnology we are handling mainly at the DNA, RNA and protein sequences, etc. So this restriction endonucleases are enzymes which are used to cut the DNA at specific regions, and this restriction endonucleases are of different types. EcoR1 is the common restriction endonuclease which is used. EcoR1 is isolated from the Escherichia coli bacteria. That is what microbes. From the microbes only we have isolated plasmids. From the microbe only we got Taq polymerase. From the microbe Escherichia coli we got EcoR1, that is a restriction endonuclease. Then the restriction endonuclease, and these are the three main components which is used in biotechnology laboratory condition, that is the plasmid, then Taq polymerase, restriction endonuclease. Now they are used in, I said they are used in, till now we learned that they are used in agriculture, they are used in industries, they are used in food technology field, they are used in medical field, etc. Then the application of microbiology in strategic fields, that is Strategic planning of a country is aimed at destabilizing another country's economy. For example, we now certain now in everyday news people say this COVID pandemic is because of a microbe which is synthesized by a country, and it's true or not, it's not yet proven. That is, strategic field is that is a strategic planning of a country is aimed at destabilizing. When two countries want to fight each other, they can use bio weapons. One example is plague. We have used it before also, plague, but it takes 7-8 years to identify whether that microbe was a what bioweapon or not. So, in strategic fields means a strategic planning by a country is aimed at destabilizing or destroying another country's economy, morale, or military affairs. For example, and a recent situation itself, if we look into, many people has lost their jobs. Many countries' economic affairs has come down because so many industries are not functioning. So that will affect a country's economy by using this bio weapons. That is known as bioterrorism. We have learned that in the previous sessions too. Bioterrorism is the usage of virus, bacteria, fungi, or toxins from a living organism to cause death or disease in humans, animals, and plants. So bioweapons or in bioterrorism, what we use is bioweapons. Usually weapons are what? Gun, sword, shield, then knife, etc. can be said as considered as a weapon. Bioweapon means biological weapons. Biological weapons are viruses, bacteria. See, it's very they are very tiny organisms. Nobody even noticed from where they came and what they are doing, and if they spread in an uncontrolled manner, people it will be beyond the control of humans. That is a dangerous situation of using bio weapons. You cannot limit, for if you are fighting with a country with a missile, or you can say, oh, 100 people will be dead like that. But bioweapons or bioterrorism, it's very, very dangerous. But these microbes, if they are left open into the field, they will multiply very fast. They have a very short multiplication phase. They multiply very fast and it will destroy many humans, animals, plants, etc. So bioweapons are used in strategic warfare. Strategic warfare means a country using bioweapon to destroy or distract or reduce another country's people, and that is what is known as bioweapons. And the common examples are black death, then smallpox, plague, etc. The main advantage is low cost. Bioweapons, the main advantage is low cost, silent, it is invisible, and it is orderless. Then many genetically modified organisms are also used as bioweapons, but it is a very dangerous condition. And genetically modified organisms are used as bioweapons, and we have learned that in previous session too. That is class A, class B, class C bioweapons. So with this, I am concluding that is we have learned about the application or usage of microbes in industrial, medical, the biotechnology, and in strategic fields.